Hello, this is Julie Venners with another Lupus Foundation of America research update. Scientists have been trying to unlock the mysteries surrounding the causes of lupus. There were a number of studies presented at the 2009 American College of Rheumatology annual scientific meeting that looked at the possible genetic basis of lupus. New technologies allow researchers to conduct powerful scans of the genome in people with lupus. They then can compare those findings to individuals who don't have lupus to try to determine what genes might be responsible for the disease. Dr. Lindsay Criswell, professor of medicine at the University of California, San Francisco, presented a review of the current status of lupus genetic research.
Compared to the 2 or 3 genes that had been discovered in the first couple of decades, during which time there were genetic studies in lupus, we now know about 24 genes that play an important role in lupus. So we're hoping that through this new information, these new genetic discoveries, we'll be able to really focus research on the underlying causes, develop therapies that can target those problems in lupus, and then develop much more effective treatments. We're convinced that genes determine to a great extent whether you have mild or severe disease. Now, if we can determine right up front what type of disease you're going to have, instead of waiting 2 or 3 years after damage has occurred, then we can really focus the more aggressive treatments on just the people that are going to get severe disease.
Dr. Criswell says that in the future, this genetic information will also help doctors individualize treatment for people with lupus to improve response and minimize side effects. Within the past 18 months, 4 major studies have identified 17 regions of the human genome found in people with lupus, compared to those who do not have the disease. Dr. Robert Graham, a researcher with Genentech, presented data from a study conducted by his team, which confirmed and expanded many of these earlier findings. And he said this genetic information may help to improve treatment of lupus.
We identified 5 new regions that are associated with human lupus, and in addition about 26 regions that showed very strong levels of evidence, but that will require a little bit more work to sort out. We can use some of the variants that we discover as being associated with human disease and prospectively test them in clinical trials that are going on, and then see if these variants alone or in combination are able to predict response to therapy. And so we're already seeing one of the major breakthroughs in lupus research over the last 5 or 10 years is the discovery of what's called the interferon signature, which is this very high levels of type 1 interferon in about half of all lupus patients. And we're already seeing that many of the genes that we've identified, about a third of the genes that we've identified, can be placed into this pathway.
Dr. Graham says that we are only at the very early stages of this research and that additional studies will be needed to completely sort out the genes that may be responsible for causing lupus. It is known that certain population groups have greater risk for developing lupus and for having more serious complications of the disease. African Americans, Hispanics, Asians, and Native Americans develop lupus between 2 and 3 times more frequently than do Caucasians. Using a genetic database of lupus patients provided by the Oklahoma Medical Research Foundation, researchers from the University of Illinois at Chicago looked at African American, European American, and Hispanic American lupus patients and compared their genetic signatures to see if they could identify pathogenic variables in lupus. Dr. Corinna Weckerle led the study and reported on the data at the ACR meeting in Philadelphia.
And what we did with our patient populations, we either looked at them as high interferon or low interferon. So using all of those variables, we then looked at whether or not there was a relationship between those variables within the different populations. And what we found is that there were relationships, but far fewer than we expected. So given all those variables, we could have had 100 million possible associations, and we actually only found 36 associations. Of those associations, very few were shared by more than one population. So what we found is that different ethnic backgrounds have different pathways leading to lupus and also have different clinical manifestations of the disease. And this forces us to look at lupus differently in different populations in terms of diagnosis and therapeutics.
Genetic information can also help physicians predict which patients may develop specific manifestations of lupus. including cardiovascular disease, which is becoming a major complication in people with lupus. Dr. Brian Skaggs, a researcher with the University of California, Los Angeles, reported on research on atherosclerosis in lupus, which has provided insight into the possible elevated risk for cardiovascular disease among people with lupus. He and his team from UCLA were looking at HDL, or high-density lipoprotein, and found that some HDL actually contributes to atherosclerosis, or blood vessel damage.
And what we found was that people with lupus have significantly more of this dysfunctional or pro-inflammatory HDL than controls that are the same age. About 44% of lupus patients versus only about 7 or 8% of healthy controls. So we decided to look at monocytes to see whether this pro-inflammatory HDL played a role in making monocytes different in lupus patients compared to control patients. And in addition to lupus patients that had PIHDL and carotid plaque versus patients that didn't. What we ended up doing is we did some experiments where we looked at whether genes were expressed more or less in patients that had lupus and PIHDL and plaque versus lupus patients that didn't. And we found only a few genes that were, including one gene that's involved in atherosclerosis. It's called PDGF receptor. And what we found was that HDL caused these monocytes to move a lot faster than monocytes that were treated with just HDL, suggesting that having this pro-inflammatory HDL or this dysfunctional HDL might contribute to increased atherosclerosis by having monocytes move into plaque and cause plaque.
Dr. Skaggs said that drugs are already on the market that target genes that may be related to these monocytes. So one idea for future studies is to see if researchers can use some of these therapies to target very specific molecules in the monocyte to halt the monocyte and therefore stop plaque from forming. More information about current and experimental treatments for lupus is provided in each issue of the Lupus Foundation of America's national magazine, Lupus Now. Published 3 times per year, Lupus Now provides updates on basic and clinical research studies, lifestyle and wellness features, personal stories, and more. To sample some of these articles and for subscription information, visit the Lupus Now magazine website at www.lupusnow.org. That's all for this episode of the Lupus Foundation of America Research Update. For additional news from the 2009 American College of Rheumatology Annual Scientific Meeting, go online to www.lupus.org/ACR. And thanks for watching.